Which of the following is the correct base pairing in DNA? A. Adenine uracil. B. Cytosine thymine. C. Adenine thymine. D. Guanine thymine. The correct answer is C. In DNA, adenine pairs with thymine, AT, and cytosine pairs with guanine, CG. Which enzyme is responsible for the synthesis of RNA during transcription? A. DNA polymerase. B. RNA polymerase. C. Ligase. D. Topoisomerase. The correct answer is B. RNA polymerase synthesizes RNA by reading the DNA template strand during transcription. What is the primary difference between RNA and DNA nucleotides? A. RNA contains deoxyribose, while DNA contains ribose. B. RNA has thymine, while DNA has uracil. C. RNA contains ribose, while DNA contains deoxyribose. D. RNA has a double-stranded structure, while DNA is single-stranded. The correct answer is C. RNA nucleotides contain ribose sugar, whereas DNA nucleotides contain deoxyribose sugar. In RNA, uracil replaces which nitrogenous base found in DNA? A. Adenine. B. Thymine. C. Guanine. D. Cytosine. The correct answer is B. In RNA, uracil, U, replaces thymine, T, and pairs with adenine, A, during base pairing. Which of the following is not a type of RNA involved in protein synthesis? A. mRNA. B. tRNA. C. rRNA. D. snRNA. The correct answer is D. snRNA is involved in RNA splicing, while mRNA, tRNA, and rRNA are directly involved in protein synthesis. Which of the following statements best describes a codon? A. A sequence of two nucleotides that codes for an amino acid. B. A sequence of three nucleotides that codes for an amino acid. C. A protein involved in DNA replication. D. A segment of DNA that regulates gene expression. The correct answer is B. A codon is a sequence of three nucleotides in mRNA that specifies a particular amino acid during translation. What type of bond holds the two strands of DNA together? A. Covalent bonds. B. Ionic bonds. C. Hydrogen bonds. D. Peptide bonds. The correct answer is C. Hydrogen bonds between complementary nitrogenous bases hold the two strands of DNA together. Which of the following enzymes is involved in the unwinding of DNA during replication? A. Helicase. B. DNA polymerase. C. Primase. D. Ligase. The correct answer is A. Helicase unwinds the DNA double helix by breaking the hydrogen bonds between the two strands. Which type of RNA carries the genetic information from DNA to the ribosome for protein synthesis? A. tRNA. B. rRNA. C. mRNA. D. snRNA. The correct answer is C. mRNA, messenger RNA, carries the genetic code from the DNA in the nucleus to the ribosome for translation. What is the function of tRNA during translation? A. To carry amino acids to the ribosome. B. To synthesize the mRNA. C. To form the peptide bonds between amino acids. D. To provide the template for protein synthesis. The correct answer is A. tRNA, transfer RNA, carries specific amino acids to the ribosome, where they are added to the growing polypeptide chain. Which process converts the information in mRNA into a sequence of amino acids? A. Transcription. B. Replication. C. Translation. D. Splicing. The correct answer is C. Translation is the process by which the sequence of codons in mRNA is converted into a sequence of amino acids to form a protein. Which base is not found in RNA? A. Adenine. B. Guanine. C. Thymine. D. Cytosine. The correct answer is C. RNA contains uracil instead of thymine, which is found in DNA. Which enzyme is responsible for adding nucleotides to the growing DNA strand during replication? A. Ligase. B. Primase. C. DNA polymerase. D. Topoisomerase. The correct answer is C. DNA polymerase adds complementary nucleotides to the growing DNA strand during replication. Which of the following structures is not involved in the process of transcription? A. Promoter. B. RNA polymerase. C. Ribosome. D. DNA template. The correct answer is C. Transcription involves the DNA template, RNA polymerase, and promoter, while the ribosome is involved in translation. What is the role of the ribosome during translation? A. To synthesize mRNA. B. To catalyze the formation of peptide bonds. C. To unwind the DNA double helix. D. To transcribe RNA. The correct answer is B. The ribosome catalyzes the formation of peptide bonds between amino acids during protein synthesis. Which of the following best describes the central dogma of molecular biology? A. DNA is replicated, RNA is transcribed, and proteins are synthesized. B. Proteins are transcribed from RNA, which is translated from DNA. C. DNA is transcribed into RNA, which is translated into proteins. D. RNA is transcribed from proteins and translated into DNA. The correct answer is C. The central dogma describes the flow of genetic information. DNA is transcribed into RNA, and RNA is translated into proteins. Which of the following components are necessary for DNA replication? A. RNA polymerase and tRNA. B. DNA polymerase and dNTPS. C. Ribosomes and mRNA. D. Ligase and snRNA. The correct answer is B. DNA replication requires DNA polymerase to synthesize new strands and dNTPS, deoxynucleoside triphosphates, as building blocks. Which of the following is true regarding the direction of DNA synthesis? A. It occurs in the 3 to 5 direction. B. It occurs in the 5 to 3 direction. C. It occurs in both directions simultaneously. D. It depends on the strand being synthesized. The correct answer is B. DNA synthesis always occurs in the 5 to 3 direction, with new nucleotides added to the 3 end of the growing strand. Which molecule serves as the template for mRNA synthesis? A. tRNA. B. rRNA. C. DNA. D. Protein. The correct answer is C. During transcription, DNA serves as the template for the synthesis of mRNA. Which of the following statements about introns is true? A. Introns are sequences that code for proteins. B. Introns are removed during RNA splicing. C. Introns remain in the final mRNA transcript. D. Introns are translated into proteins. The correct answer is B. Introns are non-coding regions of RNA that are removed during RNA splicing to produce the final mRNA transcript. Which molecule is responsible for the proofreading function during DNA replication? A. RNA polymerase. B. helicase. C. DNA polymerase. D. Topoisomerase. The correct answer is C. DNA polymerase has proofreading activity, which ensures that the correct nucleotides are added during DNA replication.